Hello, my name is Jeff Chilton, and I'm the president of Nammex. Today I'd like to talk about mislabeling and adulteration of functional mushroom products. A major issue associated with mushroom-based dietary supplements and functional foods is their extreme variability. And this is exemplified by the widespread production of products using the vegetative body stage of the fungal organism, and this is called mycelium, rather than the actual mushroom, which is the fruiting body stage. So this situation has led to a complete breakdown of proper labeling of what we call the plant part. Now, according to Professor S.T. Chang, father of the medicinal mushroom movement, speaking about the issue of quality control, he has said the active ingredient components of the majority of present commercial products have not been indicated. And he says this in a critique of what's on the marketplace. And he also goes on to say the consistency of the extracts, both in terms of overall chemical composition and in the actual levels of active components between different batches, could be standardized on the basis of one or two of the main active constituents. This is essential for ensuring some degree of uniformity in prescribed doses. Now, Professor Chang has been challenging the industry over quality control for around 30 years. In 2015, I wrote a white paper, and in that paper I defined the fungal plant parts, I identified the major active compounds, I presented practical analytical methods for the very first time, including the megazyme test for fungal beta-glucans. And in my study, I tested 95 samples in total: dried mushrooms, mushroom extract powders, and retail mycelium-based products that I purchased on the internet. This study resulted in important industry awareness of product label issues and revolutionized analysis of active compounds in functional mushrooms. Any analytical fingerprint should be based on the primary active compounds identified by scientific research and which occur in sufficient amounts to allow their use as a quality control marker. First off, and most importantly, we have the 1316 beta-D glucans, and they are utilized for immune system potentiation, and they make up approximately 50% of the cell wall of all mushrooms and mycelium. Ergosterol is the fungal equivalent of our cholesterol, and it's present in all mushroom cell membranes. So it is also present in all mushrooms and mycelium. Ergothioneine is a naturally occurring amino acid and powerful antioxidant. Now, mushrooms contain some of the highest levels of all foods. Triterpenoids occur in many polypore species, especially Reishi and Chaga. And finally, alpha glucans. We measure these because that will unmask carriers like maltodextrin, starch, and grains. Mushrooms and mycelium contain no starch, only a very small amount of glycogen. So what actually is a medicinal or functional mushroom? And we first have to define the morphological parts. Now, according to the U.S. Dietary Supplement and Health Act of 1994, DSHEA, it is a requirement for nutritional supplements that the plant part is displayed on the label. And in fact, the FDA also has a compliance document for mushroom products that states a mycelium cannot be labeled and sold as a mushroom because they are two distinct stages of the fungal organism. This is the compliance document, and it was created in 1976, and it clearly states mycelium has an identity different from mushrooms, and food products in which it is used should be labeled to clearly indicate that they contain mycelium. it also says labeling should not suggest or imply that the food contains mushrooms, since that name, by long consumer understanding and usage, is preempted by soup containing real mushrooms. Now, before we go any further, we have to understand more about this fungal organism and exactly what the different stages or plant parts are. Mushrooms do not have seeds, they have spores. These are the reproductive body. These spores land on the ground, they land in wood. When conditions are right, they will germinate into a very fine filament that we call a hypha. When multiple of these hypha come together, they will form a network, and that network is called mycelium. And mycelium is the vegetative body of this organism. And now it amasses nutrients as it breaks down the organic matter which it is embedded in. And when conditions are right, it will form a hyphal knot, a primordium, a pinhead, and then other stages of this mushroom. And the mushroom is the fruit body. And once the mushroom is mature, it will have gills on the underside that will produce spores. And now we have a completion of the life cycle. And so we have spore, which is the reproductive body. We have mycelium, which is the vegetative body. And then we have mushroom, which is the fruiting body. Now, there are actually some companies that get confused about this and call a fruiting body part of the mushroom, but the fruiting body and mushroom are the same. So what fungal parts do we actually sell as a functional mushroom? Well, in China, they grow and harvest reishi mushroom spores and sell spores as a product. Mushrooms, they are what have been used traditionally for functional purposes and for food. Mycelium is also sold as a functional product, and in China they manufacture thousands of tons of mycelium that they manufacture through a process of liquid fermentation, and these products are pure mycelium products. Mycelium is also grown out on sterilized grain, and in the mushroom industry we call that grain spawn because, again, mushrooms do not have seeds, so what is used to grow mushrooms is actually live mycelium. Now, some companies in the United States will take this grain spawn or myceliated grain biomass and they will dry it, grind it to a powder without removing any of the grain, and they will actually sell this as a mushroom product. So how do we label this myceliated grain? Is it a mushroom? Is it mycelium? Or is it actually tempeh, which you can see here in this photo? Now, in the US, myceliated grain is often mislabeled as a mushroom, and some companies even claim this product is fermented mushroom, but in actuality, it's actually fermented grain. So this myceliated or fermented grain is actually fungal tempeh, and tempeh is a food product that consists of cooked soybeans colonized and fermented by fungal mycelium, and that would be Rhizopus oryzae. Now, since grain is an integral part and a dominant ingredient, it should be listed in the supplements facts panel rather than in other, as is sometimes the case. In the list of ingredients, it should also be listed first because it is the dominant part of this product. Now, if not, is the grain an adulterant? If the grain is not revealed, it would have to be adulterating this fungal product. Now, here are examples of 3 properly labeled tempeh products. And tempeh is fermented soybeans, but it has its own very specific name. Tempeh ingredients are listed by weight in descending order of predominance. All 3 products list soybeans first, then the fungal culture, which is responsible for the fermentation. And in the EU, myceliated grain products are considered novel foods and must go through a very extensive approval process. In 2016, I brought this labeling issue to the attention of the executive director of AHPA, American Herbal Products Association, who convened a series of meetings with stakeholders, in other words, a number of companies that were producing these myceliated grain products. And the result of these meetings was a guidance policy that was put out by AHPA. However, no company making these products is currently following the guidelines, and no company is properly listing the ingredients in a manner that is consistent with regulations, similar to tempeh products, or this AHPA guidance policy. Now, the policy states each fungal dietary ingredient included in the dietary supplement is identified by its common or usual name, by the part or parts of the fungal ingredient present, and in order of predominance by weight. It also states that the parts of the fungal ingredients are the stages of the fungus present. And further down it states ingredients other than dietary ingredients in such products are disclosed. And for example, the specific substrate on which the fungal ingredient is grown. Now, how can we confirm the contents of a commercial product? And this is where we really get into testing and how we can actually determine what is in one of these products, any one of these products. Now, we use the Megazyme test, and the Megazyme test has just absolutely been a game changer in the medicinal mushroom industry. And the reason why is it is specific to mushrooms. It will identify 1,3,1,6 beta-glucans, does not measure grain beta-glucans like oats, measures alpha-glucan, which are the starches, which would be the adulterants in these products. It's accurate and repeatable. It's peer-reviewed and validated. You can train your in-house staff. It's now become a research and industry standard. Now, just to reiterate, beta-glucans make up 50% of the cell wall of a mushroom or mycelium, and the beta-glucan content will provide a clear fingerprint for every species, and lack of beta-glucans essentially indicates a lack of fungal tissue, so it's really a great test. Now, mushrooms do not contain starch. They have small amounts of glycogen, and starch, or the alpha-glucan that gets picked up, indicates the presence of an adulterant or other material, such as residual grains. Now, what this has done is it has revealed that a polysaccharide test will simply report alpha-glucans or starch as well as beta-glucans, which means the polysaccharide test, a standard polysaccharide test, is absolutely of no use when trying to analyze mushrooms. Now, this Megazyme test was actually peer-reviewed in the Journal of AOAC International in 2016. So this is a peer-reviewed method, and it was actually reviewed in the a journal of the association that actually sets quality control standards for any kind of testing. Now, our tests in my white paper showed that mushrooms and mushroom extracts have a consistent range of beta-glucans and alpha-glucans, and what we found was that the lowest amount of beta-glucans in the species that we identified was approximately 25%, and the highest was as much as 66%. The amount of alpha-glucans in these mushrooms was from 0.1 to approximately 6%. When we tested myceliated grain products, it was the exact opposite, and what we showed were very low beta-glucan values and very high alpha-glucan values. So in these products we had alpha-glucans that were from 24 to 70% of the product, and beta-glucans from 0 to 13% of the product. So they were literally the complete opposite of what you'd expect to find in a real, I should say, mushroom product. Now, another compound that works very well for mushroom analysis is ergosterol. It's a definitive marker, and you can actually use it to assess the quantity of fungal matter in a functional mushroom product. And what we found was that for mushroom samples, it was approximately 0.1% on a dry weight basis, whereas myceliated grain products were essentially a tenth of that amount. Now, this is analysis that I think is very, very important, because what we did here is we analyzed a Cordyceps militaris mushroom, and we analyzed that with approximate analysis. So we've got the fat, protein, carbohydrate, and then we also add in the beta-glucan and the alpha-glucan. And then we tested 3 different myceliated grain products and the actual grain that those products were grown on. And you can see from this that they all start pretty close there in terms of fat, but when you get to protein, they're all way off on the protein scale. In fact, it's very, very low. And then the carbohydrates in all of them are very, very high, very elevated over what an actual Cordyceps mushroom would be, which is about 55% or so. And these are all up around 70 to 80% carbohydrate, which is something you would expect, being so much grain in there. And then again, when we talk about beta-glucans, you can see that these myceliated grain products had next to no beta-glucan and very high alpha. But what's important to look at here is that these myceliated grain products track the grain that they grow on perfectly. So essentially, that fermentation process has not changed the nature of those grains in any way. So this demonstrates very clearly that what you're getting with those products is mostly grain powder. Now, in 2017, U.S. Pharmacopoeia did a very detailed study of reishi mushroom supplements. And in this study, they used 19 samples of reishi mushroom supplement products that they purchased on the internet, and then they put them through 5 different very detailed tests. The first test was a calorimetric assay of polysaccharides using iodine, and what they found was starch-like polysaccharides detected in 13 of their 19 samples. They did a saccharide mapping of polysaccharides using PACE, and that's a real detailed type of analysis. And in this, they found that there was a lack of 1,3,1,6 beta-glucans in 14 of the 19 samples. Then they did a GCMS fingerprint of the polysaccharides, and again, Ganoderma polysaccharides were lacking in 13 of 19 samples. Then they measured the molecular weight of the polysaccharides, and 14 of 19 samples were inconsistent with standard weights. And finally, they did an HPTLC fingerprint for triterpenoids, which is a marker compound for reishi, and they detected triterpenoids in only 8 of 19 samples. Now, what they stated in their results was that the results showed that the measured ingredients of only 5 tested samples, or approximately 25%, were in accordance with their labels, which suggested the quality consistency of Ganoderma lucidum dietary supplements in the U.S. market was poor, which should be carefully investigated. And what's very interesting about this study is that the vast majority of the products in this study were myceliated grain or contained starch carriers. However, every single product was called mushrooms, and so in this study, they essentially just labeled each one of those samples according to what it said on the product label, which in many cases was mislabeled and false. Shortly after the USP study came out, the American Botanical Council, in its Botanical Adulterants program, did a review of that Reishi study. And here's what they had to say. In addition, some Reishi products labeled to contain mushroom may have been made of mycelium. Mycelia are often grown on grain containing substantial amounts of starch, which could partially explain why starch was found in 13 of the 19 samples. The fact that only 26.3% of the tested products were found to be authentic suggests an immediate need for improved quality control measures. While the authors acknowledge that the sample size in this study is limited, they indicate that these 19 products almost represented the Ganoderma lucidum dietary supplements available in the U.S. market. For many years, I've been saying that a lot of the mushroom research has taken place with uncharacterized samples, and that makes it very difficult for any other scientists to replicate the studies that are being done, and that's something that's really unfortunate. And when you have products as well that are in the market that are actually myceliated grain rather than actual mushroom, that just compounds the difficulty for researchers. And this paper that was published in the International Journal of Medicinal Mushrooms is a classic example of what happens. And in this paper, they studied the correlation between TLR2 agonist activity and TNF-A induction in macrophage cells by different medicinal mushroom products. And they essentially put these products into 2 different categories. One of those product categories they called ground mushrooms, and the other category they called mushroom extracts. Now, to their credit, and something that's not normally published, is the names of the actual products that they were testing. And looking at those names, I was able to see immediately that 9 out of 10 of the products that they were calling ground mushrooms were actually myceliated grain products. So they were unable to tell the difference due to the label. So we have scientists here trying to do an actual study using commercially sourced products, and they were unable to actually know what they were utilizing, completely uncharacterized. This is a situation where they are basically in this paper saying that dried mushroom products, this is their activity compared to extracts, when that was not the case at all. It had nothing to do with dried mushroom products. Now, I am a member of the International Society for Medicinal Mushrooms, and this is their journal, so I immediately wrote up a paper to essentially outline what went on here. And essentially what I stated was a recent study published in this journal demonstrates the pitfalls faced by researchers who utilize commercial products as their test samples without proper characterization. Labeling of commercial mushroom products is often incorrect, which can lead to erroneous interpretations and conclusions. 9 of the 10 samples of commercially branded products used in this study and identified as ground mushrooms were actually grain spawn, mycelium propagated on grain. So fortunately, the paper was published, and I'm hoping that that will alert researchers to the fact of the need to be so very careful when you're bringing in commercial products into your research. In 2016, I presented my analytical fingerprinting method to the conference sponsored by the International Society for Mushroom Science. And at that point, I was using beta-glucans, alpha-glucans, and ergosterol, and using a mushroom as the baseline for evaluating any type of product, whether it was a commercial mushroom product or just a mushroom powder that somebody might submit. In 2019, I presented my fingerprinting method to the 10th International Medicinal Mushroom Conference, and at that point in time I had upgraded the method to include ergothioneine, and I had tested 160 samples with which to put together an actual very robust fingerprint for each one of the major medicinal mushroom species. So using the analytical testing data of 4 major constituent markers in functional mushrooms, we were able to produce a standardized fingerprint and establish it for every mushroom species that we sell. And again, based on beta-glucans, alpha-glucans, ergothioneine, and ergosterol. And this fingerprint can be used to compare against unknown products. So it's really a very important tool for quality control with any particular business that is selling medicinal mushrooms. Now, the other part of this is that you can add additional compounds to this fingerprint, as we have done, for example, with reishi triterpenoids, or in fact with cordycepin from cordyceps species. In 2015, Professor S.T. Chang actually referenced my white paper at the first Chinese Ganoderma conference. And in his talk to this group, he basically reiterated mushroom, mycelium, and spore are 3 different concepts. He also states mycelium is the structural organization of fungal organisms, and mushroom is the fruit body. The medicinal value of fruit bodies has been confirmed. He also says, from the medicinal value perspective, Basidiomycelia produced using grain as a culture substrate contains a small amount of beta-glucan, a large amount of starch, and a small amount of secondary metabolites, including triterpenoids, sterols, etc. In contrast, fruit bodies contain a lot of secondary metabolites as well as plenty of beta-glucan. Therefore, Professor Chang said that the fruit body is the truly comprehensive biological stage of basidiomycetes, and the Ganoderma fruit bodies have the real medicinal values. I was first introduced to Professor Chang in the late 1970s when I came across his book, The Biology and Cultivation of Edible Mushrooms, of which he was an editor along with Professor Hayes, both of whom had chapters in this book. This book was a very, very important part of my understanding of mushroom cultivation as well as medicinal properties. And later in the 1990s, when I traveled extensively in China, I had the opportunity to meet Professor Chang at a number of different mushroom conferences. In 2019, at the International Society for Medicinal Mushroom Conference, I also was able to see my old friend again after so many years, and I just want to say that he has been an inspiration, and he has been someone who has spoken out for years about quality control of medicinal mushrooms, and I'm so happy to be able to contribute to that cause. In summation, in order to increase and maintain consumer confidence, protect public health, and to meet current and future quality demands, as well as safety criteria set by regulatory authorities, accurate labeling of fungal parts is essential. Only by product testing can labels be accurately assessed. Testing of beneficial compounds is also critical for ensuring product quality and a degree of uniformity in prescribed serving sizes. For more information about the Nammex testing protocol, my white paper is available on our website. Thank you very much.